Hello, my name is Aryan Shenoy, and today I'll be telling you about the hybrid topological neural network, a novel approach to signal segmentation for the optimization of the regenerative peripheral nerve interface. 185,000 individuals undergo amputations annually in America alone, and one would think with modern medicine and technology, these people would receive the best treatment possible through prosthetics. However, this is not the case. Up to 50% of amputees report not using their prosthetic devices due to a lack of intuitive control, fine motor control, and refined sensory feedback. In 2016, my mentor and his team developed a device known as the Regenerative Peripheral Nerve Interface, or RPNI. This device is constructed of transected peripheral nerves and a bioamplifying muscle graft. While originally created to combat neuroma pain, 4 years later it was discovered that different regions of the RPNI corresponded to different degrees of freedom in the hand. The lab theorized that by placing electrodes on the RPNI, they would be able to control a prosthetic device, and they were right. Later, the lab wondered how they could improve control of a prosthetic device through the RPNI. And the solution to this was to create multiple RPNI devices to capture as many degrees of freedom in the hand as possible. However, this poses another challenge, as the surgery for a single RPNI device is invasive, and by adding more and more devices, the recovery is even more painful. The solution to this is a single RPNI device containing multiple peripheral nerves. However, due to the proximity of the nerves and the presence of the muscle graft, it is impossible to differentiate between the independent control signals generated by the nerves. Standard signal segmentation techniques such as principal component analysis, independent component analysis, and wavelet transform all have individual strengths but pose significant shortcomings when applied to neural signal segmentation. All of these factors led me to my problem, which is when multiple nerve fibers overlap in a single RPNI device, the origin of the independent control signals is unknown. The goal for this project was to create a signal segmentation algorithm that can outperform traditional strategies by achieving accuracy of more than 95% while not compromising performance or efficiency. The project began with data labeling and preprocessing, a crucial step to ensure the quality and accuracy of the machine learning model. The dataset used in the study was collected over an 8-year period by my mentor, during which extensive experiments were conducted on RPNI-based signal acquisition. The dataset consisted of raw EMG recordings, which were then labeled according to 13 distinct classes, each corresponding to a specific degree of freedom in hand movement. Since EMG signals are inherently noisy due to artifacts from muscle contractions, electrode placement variability, and environmental interference, a bandpass filter was applied. This filter effectively removed unwanted low-frequency motion artifacts and high-frequency electrical noise while preserving the critical signal components needed for neural decoding. Next, feature extraction was performed using a hybrid approach that leveraged both time domain and frequency domain characteristics. The temporal features, including mean absolute value, root mean square, and zero crossing rate, were chosen because they effectively capture muscle activation patterns over time. Meanwhile, spatial features, derived from power spectral density and dominant frequency components, provided insight into how different nerve signals contributed to overall motor intention. The combination of these two domains created a richer representation of the signals, offering both fine-grained detail as well as a broad global structure. The topological portion of the project posed the greatest challenge, as it required transforming the extracted features into a multidimensional point cloud. Each feature was plotted in a high-dimensional space where similar signals clustered together while dissimilar signals remained apart. Given the complexity and size of this data, dimensionality reduction was necessary. To reduce computational load while retaining critical information, principal component analysis was applied. This transformation condensed the high-dimensional feature space into a lower-dimensional form while maintaining the most relevant variations between different signals. Once the dimensionality was reduced, a cyclical complex construction was performed. This method connected the reduced data points into topological structures, creating shaded regions within the multidimensional space. These regions represented clusters of related signals, ensuring that distinct neural activities were preserved in a mathematically meaningful way. The final topological step was applying persistence homology, which helped analyze the structural features of these regions. By tracking how different features appeared and disappeared across multiple dimensions, persistence homologies allowed the system to distinguish overlapping signals and map them back to the correct hand movement class. At this stage, a neural network was implemented to refine and automate the segmentation process. A standard feed-forward neural network architecture was selected due to its efficiency in processing structured data. The network utilized an atom optimizer to dynamically adjust learning rates and improve conversion speed, while a ReLU activation function introduced nonlinearity to enhance pattern recognition. The network architecture consisted of multiple layers that progressively distilled the input features into a final classification vector. The final output layer produced 13 possible class activations, with the highest confidence prediction serving as the final output. As expected, the findings for this study are both promising and exciting. The model minimized the amplitude error to 0.02 as seen in the consistent overlap in this figure. The greatest error was seen during the nonlinear portions of prediction, however my model was easily able to cope with this issue. In the time domain, my model achieved a temporal precision of 99.7%, while maintaining an average onset latency of a measly 2.1 milliseconds, which is incredible when compared to the CNN baseline, which at times was off by as much as 1.5 seconds. My model was also very successful in utilizing topological data analysis, as all the classes were well above the confidence threshold as seen in the figure on the left. And the model had an average Wasserstein distance of 0.1, meaning that the shaded regions within the multidimensional point cloud were far enough apart that there was no overlap or confusion. Finally, my model was consistently matching the CNN baseline in power consumption, demonstrating a power spectral density similarity of 0.95, suggesting that the model would not generate additional latency despite its complexity. There were several notable takeaways from this project, one being the accuracy of 97.8%, which suggests a robust generalizability across EMG classes. Additionally, I found uniquely that deeper layers of the HTNN enhance signal isolation, which is likely due to the distillation workflow. Another interesting discovery was that certain mid-range frequencies, as well as controlled noise, improved accuracy and reduced classification times. Finally, the structure and performance of my model suggest use cases in other types of signal segmentation methods. To wrap things up, the problem I tried to address with my research is that overlapping nerves cause issues in neural signal segmentation. The goal for this project was to create a system that can accurately segment these signals without creating performance problems. And by applying dual feature input, topological data analysis, and machine learning, I was able to more than achieve this goal. This project has the potential to better the lives of 57.7 million global amputees. I'd like to remind you that a staggering 50% of amputees end up not using their prosthetic devices due to issues that my model can easily alleviate. I hope that by implementing this system, more patients will receive the high-fidelity, real-time adaptive treatment that they deserve, and that some of their pain and recovery can be reduced. In the future, I wish to validate my findings in the study by applying the system to real patients. Additionally, I want to look at other types of prosthetic devices as this project focused solely on upper limb devices. And finally, I want to look at the HTNN use cases in other techniques such as surface electromyography that suffers from similar problems to the regenerative peripheral nerve interface. My name is Aryan Shenoy, and it has been a pleasure sharing my work with you. Thank you.